Let me introduce you to the DNA molecule that makes up our genes. DNA is made up of molecules called nucleotides. Each nucleotide contains a phosphate, sugar, and a nitrogen base. Different types of nitrogen bases are thymine, adenine, guanine, and cytosine. The nucleotides create two strands attached to each other by bonding between bases. The strands twist and create the structure of DNA double helix. DNA replication is a complicated process of copying the cell's DNA that occurs in the nucleus. It happens during interphase and a lot of enzymes are involved. It begins when helicase starts to unzip the strands by breaking the hydrogen bonds between the base pairs. Now we have got two separated strands that will be a template for the new ones. These parental strands are called a lagging and a leading strand. According to orientation of the sugar carbons, the strand is either lagging and it's oriented 5' to 3', or leading when it's oriented 3' to 5'. As a result of this, the strands are replicated differently. First, let's talk about leading strand. An enzyme called primase produces a short piece of RNA called primer, and that then binds to the end of the leading strand. The process is easier because polymerase, the enzyme that creates a new strand, can only work in 5' to 3' direction, and leading strand is oriented like that. Now the polymerase knows where to start adding new nucleotides. When it comes to replication of the lagging strand, things get a little bit more difficult. The polymerase can move just in 5' to 3' direction. So as we've mentioned before, so the new strand can be done all at once. Primase makes many RNA primers that then bind at various places along the lagging strand. Then the polymerase do its job. It makes chunks of DNA called Okazaki fragments in the 5' to 3' direction. The problem with this is that the new strand is still made up of RNA primers, and these have to be replaced by nitrogen bases. The exonuclease enzyme puts away the primers, and the gaps are filled by ligase. Fortunately, the polymerase enzyme rarely makes mistakes, but if it occurs, it can lead to, for example, mutation. The replication is called semi-conservative because it creates two DNA molecules consisting of one old and one new chain of nucleotides. After replication, DNA automatically rolls into the double helix. That's all.